Hello everyone, you are welcome to my YouTube channel. I'll be discussing the topic trichinosis. Trichinosis is also known as trichiniasis. It is caused by Trichinella species. There are 12 recognized taxa which are genetically and biologically delineated into two distinct clades. The presence or absence of an intramuscular collagen capsule is used to characterize these 12 recognized taxa. You have the encapsulated clade, which include the following: Trichinella spiralis, Trichinella nativa, Trichinella genotype T6, Trichinella brevoorti, Trichinella genotype T8, Trichinella murrelli, Trichinella nelsoni, Trichinella genotype T9, and Trichinella T12. While the non-encapsulated clades include Trichinella papae, Trichinella pseudospiralis. Briefly, the life cycle of Trichinella species. The same animal can act as definitive host as well as intermediate host. Definitive host, that is, the sexually matured adult female and male Trichinella species can be found. And also as intermediate hosts where the larval stage can be found. All stages in the life cycle of Trichinella are found in individual mammalian hosts. When inadequately cooked meat, skeletal muscle containing the infective larvae is ingested by another mammal. The larvae are released by the action of gastric fluids and they pass into the small intestine, where they invade the epithelial wall of the small intestine, mold 4 times, and becomes sexually mature. After copulation, the females begin to expel the newborn larvae, known as embryo, about 6 or 7 days after infection. There is evidence that they may survive for much longer periods, especially if the host immune system is compromised. Most of the newborn larvae penetrate into the submucosa and by hematogenous spread move to various organs, including the following: pancreas, the lungs, the brain, the lymph nodes, the myocardium. The newborn larvae are enclosed in a capsule and further calcified beginning as early as 6 months or within 2 years. The calcified, capsulated, encysted larvae appear as fine opaque glands. Here is the life cycle of Trichinella species briefly. This is the human host, and also you have the domestic cycle consisting of the pig and rat. The rat can be eaten by the pig, and that keeps the cycle. And man becomes infected by eating or ingesting undercooked meat, particularly pork. So the encysted larvae in the striated muscles are released and by the gastric juices in the small intestine. Then the sexually matured, the larvae become sexually differentiated into male and female adult worms, which are found in the small intestine. Then the following copulation, the larvae are produced, which can be deposited in the striated muscles. In man, the life cycle of the parasite Trichinella ends there. That is, man becomes a blind horse. His flesh is not available to any other carnivorous animal for consumption. The sylvatic cycle ensures that Trichinella is maintained, and when man or humans decide to hunt for these animals, like the bears, the quaggas, the walruses, they also have the encysted larvae in their striated muscles, which can be released, and the cycle is completed following the differentiation of the larvae into adult male and female worms. Epidemiology of trichinosis. The outbreaks have resulted predominantly from consumption of Trichinella infected pork. Also of importance is the growing proportion of outbreaks caused by the sylvatic Trichinella species. When hunters or tourists want to have a taste of the meat of bear or quagga or walruses, so either directly through game meat or through spillover to domestic animals. Recent reports also indicate that infected herbivores, horses, sheep, goats, and cattle have been the source of outbreaks. A new variation on the traditional model of trichinosis epidemiology involves the following. Hunters and those in close association with them are at risk of contracting trichinosis or trichinosis, beg your pardon, when raw meat from game animals, for example, bears, quaggas, foxes, walruses, and wild pigs is eaten. Also, population dynamics of humans with their own food practices, including the consumption of raw meat. Equally, the illegal importation of uncontrolled meat from endemic to non-endemic countries and new food practices and dishes, including raw meat, resulted in outbreaks in Denmark, Germany, Italy, and Spain. The increasing number of international travelers has resulted in many reports of tourists who acquire Trichinella infections while traveling or hunting in areas of endemicity. They just want to have a taste of something different, and in the process, they get infected. Another important source of infection related to local meat consumption habit is found in France, where most trichinosis cases have been traced to consumption of raw horse meat. In Italy, human infections due to the consumption of horse meat have been documented in 2 areas where the French habit to consume raw horse meat was introduced some centuries ago. The consumption of raw or undercooked meat is strongly associated with Trichinella infection in humans. Cultural factors such as traditional dishes based on raw or undercooked meat or meat-derived products play an important role in the epidemiology of the disease. Briefly, we'll look at the clinical manifestations of trichinosis. Trichinosis is one of the few helmintic diseases that often runs with consistent fever that may persist for 3 weeks. There are 2 phases of trichinosis in the human host. an enteral or intestinal phase, and a parenteral or systemic phase. In the first phase, that is the enteral phase, parasite bodies higher than a few hundred level can initially cause gastroenteritis associated with diarrhea and abdominal pain, approximately 2 days post-infection. The first gastrointestinal signs result from mucosal invasion of the first stage lab. These signs may last for 2 to 7 days, but may persist for weeks. We have the acute stage trichinosis and have the chronic stage trichinosis. The acute stage trichinosis include non-specific symptoms such as headache, fever, uneasiness, fever-associated chills, and occasionally gastrointestinal disorders. It marks the beginning of the enteral phase of trichinosis in a human host. There are also immunological, pathological, and metabolic disturbances. These are caused by the migrating larvae of Trichinella and their metabolites during the acute stage of the infection. Infiltrating inflammatory cells such as mast cells, eosinophils, and lymphocytes, and a rather Th2-oriented cytokine profile constitute the immunological reaction. There is also periorbital edema, which occurs because of the release of histamine, serotonin, a slow-reacting substance, SRS, of anaphylaxis, bradykinin, and prostaglandins, following the increased permeability of capillaries and a leakage of fluids, electrolytes, albumins, and cell elements into the surrounding tissue. The second stage, the chronic stage, trichinosis, will include complications and secondary infections. But it should be noted that neurological complications are rather rare in trichinosis patients. But where they occur, they show multiple small subacute cortical infarcts or infarctions upon magnetic resonance imaging. Patients exhibiting such neurological complications can show consciousness disorders or excessive excitement, and also frequently somnolence and apathy. Most brain abnormalities can be visualized using the magnetic resonance imaging MRI scan or computed tomography CT scan. Laboratory diagnosis of trichinosis. Definitive diagnosis will depend on several laboratory tests, such as the intradermal test, which involves an antigen which is prepared from Trichinella larvae. A wheel of 5 mm or more diameter surrounded by an area of erythema is regarded as a positive test. Other tests include bentonite flocculation and latex agglutination tests. Muscle biopsy is usually is most widely used. A definitive diagnosis can be made when the first stage larvae are found in the muscle biopsy, generally performed in the deltoid muscle. The deltoid muscle is preferred because it's more accessible. Histological examinations may also reveal modifications of the skeletal muscles, including basophilic degeneration of the fibers, fatty metamorphosis, hyaline or hydropic degeneration, or both, and interstitial inflammation. There could also be elevated levels of muscle enzymes such as aldolase, lactate dehydrogenase, and creatine phosphokinase in blood, which indicates myocystis. These have been found in 75 to 90% of infected persons between the second and fifth week of infection. Other techniques include enzyme-linked immunosorbent assay, which is with the acronym ELISA, or polymerase chain reaction PCR. Prevention and control of trichinosis. The public should be informed of the danger of trichinosis through educational campaigns. Antihelminthics such as albendazole and mebendazole are the principal drugs for the treatment of trichinosis. To relieve muscle pain, glucocorticosteroids can be administered. The most important thing is adequate cooking of meat with the core temperature reaching 70 degrees Celsius and until the pink color is changed to gray. Most people are interested in tasting meat before it is done. Focus and leave it, let the color change from pink color to gray. Then you are sure that at least the meat is properly cooked. Freezing of meat at minus 20 degrees Celsius for at least 3 weeks will ensure that the larvae of Trichinella species are become inactive. And where it is permitted, irradiation of meat can be used. Here are links for further reading. Thank you for listening.